and welcome to the BEaTS Science Seminars Online. Good morning, and thanks for being here with us today. Just a little housekeeping before we get started with our seminar. The seminar will be a 30 to 40 minute uninterrupted presentation by our speaker today, and it'll be followed by about 10 minutes of questions. If you can keep your microphones off and your camera also off during the seminar, that would be great. If you get disconnected for any reason, the meeting ID is 477-236-347. So for all of your questions that you have during the seminar, you can send them to me in a private chat, and I will ask Dr. Willerth the questions after the presentation. If you don't mind including your name as well as your institution that you belong to, maybe the program and your affiliation, that would be nice. And after the seminar is done, the seminar will be available on the BEaTS Research Radio YouTube channel about a day after. So in about 24 hours, you'll be able to rewatch the seminar today. So today for our seminar, we have Dr. Stephanie Willerth with us from the University of Victoria, speaking to us about 3D bioprinting personalized neural tissue for drug screening. And next week, we have Dr. Ali Kedem Hosseini, who will be speaking to us about engineering and precision medicine. So don't forget to tune in at the same time next week. So our speaker today is Dr. Stephanie Willerth. Dr. Willerth is a professor and acting director of biomedical engineering at the University of Victoria. Since starting with the faculty in 2010, Dr. Willerth has successfully run an interdisciplinary research program investigating neural tissue engineering treatments for disorders of the central nervous system. Her research combines stem cells, tissue engineering, drug delivery, cellular reprogramming, and bioprinting. She has been recognized as a leader in the field of biomaterials and tissue engineering, as evidenced by her Tier 2 Canada Research Chair in Biomedical Engineering, being named a Women of Innovation in 2017, and a Young Innovator in Cellular and Molecular Bioengineering in 2015, and has served as the President of the Canadian Biomaterials Society in 2017 and 2018. And today, as I mentioned, we have the pleasure of having Dr. Willerth with us today to speak about 3D bioprinting personalized neuro tissue for drug screening. So without any further ado, I would like to invite Dr. Willerth to start sharing her screen for her seminar today.
Excellent. Okay, there we go. I would like to share my screen. Yeah, well, first of all, thanks for having me. It's a bit of a change of pace given the crazy times to actually talk about the work we were doing before everything got shut down. And I hope everyone listening is doing okay in these difficult times. Okay. All right. And thank you, Holly, for the lovely introduction. For those of you who haven't been to University of Victoria, you can see our campus right here. It's inside of a ring road. And at the University of Victoria, my lab's actually located in our medical school, which is affiliated with the University of British Columbia. I have joint appointments between our medical school and the Department of Mechanical Engineering. And I also have an affiliate appointment with the School of Biomedical Engineering at the University of British Columbia. And our group has been really productive in the last few years using 3D bioprinting, which I think is a really exciting tool for engineering all sorts of tissue. And our lab has an emphasis on engineering neural tissue using stem cells. And so I'll talk a bit about that today, and I'm happy to take any questions once my talk's over. So tissue engineering, for those of you who aren't familiar with, combines the principles of biology and engineering to develop functional substitutes for diseased and damaged tissue. It combines things like biomaterial scaffolds, drug delivery, and cell therapy in different combinations in order to create these replacement tissues. And there's a lot of a wide range of tissues you can make. Things that are quite simple, such as artificial bladders, are already in use. There's a famous Vacanti mouse where they grew this ear on a mouse to be transplanted back into people. And then for those of you who want to get distract yourself during these difficult times. I've been giving this talk, or I've been using this slide since I've started as faculty back in 2010. And one of the examples I used to give of engineered tissues being transplanted into people were these artificial tracheas. And then it turned out that Paolo Macchiarini, who was one of the doctors responsible for this work, actually had been pulling off a large scam, and they wrote a whole article up on it in Vanity Fair. And so if you're bored and want some interesting reading, you can read all about the details of his scam and sort of how he faked this data to get these engineered tissues into people. So one of the things that you don't really see when you Google for images of tissue engineering, if you look for neural tissue, you don't get to see things that look like these structures, like brains or spinal cords. You usually see these drawings, and you'll see some of the reasons why it's a bit more difficult to engineer neural tissue. But one of the things that you have to, or some of the considerations you have to think about are being able to regenerate both the mechanical properties of the cells present in your neural tissues, as well as their electrical and chemical signaling that they produce. In terms of why do we need neural tissue engineering, a lot of people know someone who's had one of these neurodegenerative diseases. Things like Alzheimer's and Parkinson's are quite prevalent. And here in Canada, there's a large incidence of multiple sclerosis. There's actually specific funding agencies, for example, Brain Canada, that are focused on trying to develop better treatments for a lot of these diseases and disorders. In terms of disorders, my PhD work had focused on looking at different ways of engineering neural tissue for treating spinal cord injury, and I am an active member of ICORD, which you can see here, with Wolf Tetzlaff, Brian Kwan, and Rick Hansen, very big research center over here in British Columbia that's dedicated to finding different ways of trying to treat and cure spinal cord injury. And there aren't really true cures for things like spinal cord injury, even traumatic brain injury. Down here in the bottom, you can see a picture of Michael Schumacher, who's possibly one of the greatest race car drivers to ever live, who suffered a tragic skiing accident, and despite his immense resources, still hasn't really been able to recover from these injuries, just to give you an idea of how devastating they are. In terms of bioprinting and motivation, one of the things for these neurodegenerative diseases and disorders is a lot of them, as I mentioned, lack true cures. You usually are prescribed drugs that will just treat the symptoms. And even then, the pipeline to get drugs tested for these different disorders is long and expensive, costing billions of dollars and taking over a decade. In the case of neurological drugs, over 90% of those going into clinical trials fail, costing a large amount of money, especially to pharmaceutical companies. And so there's a lot of interest in finding better tools for predicting whether or not a drug will be effective before it gets to the level of clinical trials. And partially some of the reasons for this include, as you can see in the next slide, and I actually wrote an invited commentary on this for the Journal of 3D Printing in Medicine about why bioprinting can potentially serve as a better tool for drug screening. And that's because animal models currently serve as the gold standard for evaluating the efficacy and toxicity of drugs. And for graduate students out there who are doing animal studies, or were doing animal studies, they are very labor intensive as well as costly. And the biology of mice and rats doesn't necessarily translate quite accurately to humans, but it is one of the models that has been commonly used and therefore used commonly in industry. Another alternative is to use human tissue slices cultured in vitro, which have better predictive ability, but have limited supplies. So what my group and many groups around the world do is look at how to engineer tissues from human-induced pluripotent stem cells. And so back in 2006, and I actually saw Yamanaka give a talk on this that year at the ISSCR meeting in Toronto, he showed that you could take an adult cell, and in this case it was fibroblasts, and you could treat them with these reprogramming factors. They would become a cell that could become any cell type found in the body. So it's quite exciting because previously to get a pluripotent stem cell or one of these stem cells that could become any cell type found in your body, you're going to have to derive them from embryos, which was a bit controversial back in the day, as well as the fact that you weren't necessarily going to have your own embryonic stem cell line, because usually there was one embryo and that's the embryo that made us. And so this iPSC, the development of these really was a powerful tool for making both patient-specific tissues, which is what I was very excited about back in 2006. But also, as I'll talk about later, this actually is a very powerful tool for modeling neurodegenerative diseases, because you can take these cells from patients suffering from diseases like Alzheimer's or Parkinson's and reprogram them back into these stem cells and then actually derive humanized tissues that have some of the hallmarks of these diseases. And in fact, it was such an important discovery that Yamanaka was awarded the 2012 Nobel Prize. And then in case you're bored in quarantine and want to learn more, I did write a book about this when I was on sabbatical at the University of Wisconsin-Madison back in 2016. So what kind of cells are we trying to produce from these induced pluripotent stem cells? So one of the most important cells in the nervous system are the neurons, which are the excitable cells that transmit information throughout your body using electrical chemical signaling. There's a lot of different types of neurons, and covering them with a myelin sheath, and this helps them essentially transmit these electric signals throughout our body. And then there's finally the astrocytes, which help provide support to the cells of the central nervous system. They also make up the blood-brain barrier, which protects the central nervous system by providing this immunoprivileged barrier. The presence of the blood-brain barrier also is potentially one of the reasons why we see such high drug failure rates. The way we've evolved is to really protect the brain and spinal cord, and this blood-brain barrier really makes it hard for drugs to cross in and cross over into the brain. I'm not going to talk about it much today, but something that I think is really interesting, and my lab has looked at a bit, is the idea of being able to create a vasculature within some of our tissue-engineered constructs. And there's some really cool work being done by Parellis, a company out of California, where they have some interesting microstructures you can purchase and then make your different types of engineered tissues around them. And then it has this vascular structure internally in your tissue. So how do we get these cells that can become any cell type found in the body to become the cells of our central nervous system? And our group has been focused on using small molecules. And these often can influence cells in a manner similar to growth factors, but they tend to be less expensive. There's a lot of small molecule screening to find out which small molecules are the most effective at promoting the differentiation of these hiPSCs into the specific neural subtypes. And I'll talk about later, we often encapsulate both growth factors and these small molecules to promote the differentiation of our tissues. And so the fact that they're less sensitive to temperature and pH makes them more attractive when you're encapsulating them for controlled drug delivery systems. And some of the common ones you'll probably hear me talk about today. There's retinoic acid, which is a vitamin A derivative that helps establish polarity during development by activating the HOX genes. This is really important in the formation of our hindbrain and neural tube during development. Another interesting small molecule that we've been able to deliver in a controlled fashion is puremorphamine. It's a small molecule agonist against the protein smoothen, which activates the sonic hedgehog pathway. So normally the signaling pathway is activated by a protein called sonic hedgehog. And this pathway ensures that you get patterning of the ventral neural tube, and in particular encourages neuronal differentiation into motor neurons. And back in 2015, one of my master's students, Amy Montgomery, she showed that we could really effectively induce neuronal differentiation of iPSCs using this molecule. And I think Sonic now has his own terrible movie that's animated. It's probably out by now. So just to talk a little about some of the issues of non-bioprinting methods of engineering neural tissue from iPSCs, differentiation protocols often take weeks and require significant amounts of labor. And this is pretty standard, not just for neural tissues, but if you talk to other groups or if anyone in the audience is deriving tissues, such as cardiac tissue, islets, and things like this, a lot of the methods for engineering tissues and differentiating your cells will take weeks. They require a significant amount of labor, media changes, and so forth. We previously used some neural aggregate cultures, and they would sort of spread out in all directions and not accurately replicate structures found in vivo. Another technique that's really interesting and has gotten a lot of publicity, although pharmaceutical companies don't necessarily like it, is you can often make organoids or mini tissues using the intrinsic potential of these induced pluripotent stem cells. And so in our case, you can do, I think, a 60-day protocol. It involves some shaking steps and reseeding and lots of media changes to get these essentially mini brains in a dish. And that's what the picture's showing here. As you can see, they look really cool. But due to the fact there's a lot of variability and only about 25% of the organoids form the structures they're supposed to, means that we need to look to alternate methods. And so I think that 3D bioprinting can help address some of these limitations, particularly with regard to reducing the amount of labor and increasing the reproducibility of our tissues. So what is 3D bioprinting? And so essentially, like normal 3D printing, you have an initial CAD file or computer-aided design file that contains the design configurations for your tissue. And then the bioprinter itself will take things like a biomaterial or a bioink containing stem cells, and then based on these specifications, it will actually print tissues in the structures that you desire. And some of the things when you are 3D bioprinting is to ensure that your cell function and viability are preserved within the construct. Just like with other traditional tissue engineering methods, you will then culture these tissues in the presence of cell culture media, and then you'll get some tissue that can potentially resemble the brain or spinal cord, which you can use for either drug testing, studying fundamental biology, or I think a bit further down the line, you could potentially be implanting. And we actually have a commentary that I wrote with Simon Bayer and Conrad Wallace, who helped co-found Aspect Biosystems, which I'll talk about in a second, that's coming out at Current Opinion in Biomedical Engineering, that essentially reviews the literature associated with bioprinting both healthy and disease models of brain tissue. So things to consider when you're bioprinting. One is the method of printing. There's a lot of different bioprinters out there on the market, and they range in cost from your very low-end Alevis all the way up to the Aspect RX1 and the bioassembly bot, which you're looking at a lot of money for. There's also some interesting mid-range options. For example, CellLink has a new product out called the BioX, which I think does a really nice job of integrating a lot of functionality at a reasonable price point. There's also what types of cells you want to print. As I mentioned, brain tissue has a number of cell types, the same for cardiac and other tissues. Another choice is you want to print primary cells versus stem cell-derived cells. And one of the things with printing induced pluripotent-derived cells is that they tend to be a bit more sensitive compared to primary cells or more standard cell culture when you're trying to bioprint things like fibroblasts or cancer cells. Another consideration is what bioink you're going to use. Bioprinting is a really interesting interdisciplinary field because oftentimes the materials that are easiest to print aren't necessarily the ones that are going to promote optimal cell survival and function. Tissue structure is another important factor. In that commentary I just mentioned, it was sort of interesting for the brain tissues. A lot of the structures that are printed are actually a lot of very simple cross-hatching, which aren't really replicating physiologically relevant structures. And that's something my group has been looking into, is trying to make tissue structures that more accurately mimic what you'd find in the brain or spinal cord. And then what factors, including small molecules or growth factors, should be incorporated into the media or tissue to ensure that we're getting the tissue formation that we desire. So there's a couple of different commonly used types of bioprinting, and so these include inkjet printing, where you can be distributing your cells in your bioink using thermal or acoustic methods. You can also dispense your bioink into the desired pattern using laser pulses, which is what laser-assisted printing is. We use a variant of extrusion printing. It's a bit more complex that you'll see in a second, but you can either use pressure or mechanical force in order to dispense your cells plus bioink into the structure that you desire. And there's also cell electrospinning, which could technically qualify as bioprinting. Just like normal electrospinning, you apply a highly charged field and you draw out these very fine, thin filaments, both consisting of bioink and your cells as well. And if you want to read more, my lab has 2 review papers, one on bioprinting models of neural tissue that came out in Brain Research Bulletin in 2019, and then one that came out in 2018 that was a collaboration with Benato Jodar's group at the University of Texas, El Paso on 3D bioprinting stem cell-derived tissues, which was one of the most downloaded papers at Cellular Molecular Bioengineering. And that one's a more general review, just about how you bioprint stem cells in general. So our method at the Willerth lab that we actually have a number of bioprinters, but the one that is the most sophisticated is we do have one of Aspect Biosystems RX1s, which has their lab on a printer microfluidic technology. And so one of the reasons this technology is so exciting, as you can sort of see here, where they've used food coloring, you can actually see how they can switch between the different colors. So you can actually have multiple inputs, so you can print multiple types of materials as well as multiple types of cells, which will give you complex structures such as the bioprinted trachea that you're seeing in the bottom that's printed in a ring with multiple colors. So one of the reasons this technology is so exciting is that the way the microfluidic print head works is that it minimizes shear on the cells, and this really enables us to get a high level of cell survival. You can print these multiple materials and multiple cell types, which, although I won't talk about it today, we do have some exciting work we're hoping to submit where we've printed co-culture models of both cancer and healthy tissue. The process is relatively quick. You can make 4 tissues in 5 minutes of printing, and in theory, it's compatible with a broad range of biomaterials. And so, as you'll see, that usually it works by having a bioink that then mixes with the crosslinker, and then it extrudes into producing your tissue. And they had a paper come out on their trachea tissue models in the FACEP journal this year, if you want to learn more. So our group and a pair of sisters, Emily, who's taking a directed study with me this summer, and Lila Abelseth, who currently serves as my administrator of my research center, along with Laura and the guys at Aspect, we published a paper demonstrating how we had a novel fibrin-based bioink. And so those of you who've worked with fibrin may know that it's a very good biomaterial for supporting neural cell growth and differentiation. My lab had spent lots of years optimizing its formulation in a 3D format in wells that was very low throughput and very labor intensive. And we were able to translate this to make a version that was printable, that really did a great job at supporting cell growth. And in this case, we were using it to print human-induced pluripotent stem cell-derived neural progenitors. And this paper came out in ACS Biomaterial Science and Engineering in 2019, and it was also featured in their Virtual Methods of the Year issue. And so it's a really nice paper showing step-by-step how you go through and make each of these solutions and how you print your cells, and validating our bioink as an important tool for engineering neural tissue. And so initially, so here's some of our initial structures that we had printed using our bioink. We were printing essentially cylinders in an attempt to replicate the spinal cord. And then we also printed some ring structures, mainly because Aspect is quite the big fans of rings. We are currently looking at printing some more complex structures. My student Ruchi Sharma had a paper come out in Frontiers in Bioengineering this year, where we actually printed half domes, which is a pretty complex structure, and we hope to build upon that work to further develop that and refine it as a model of how we want to model brain tissue in a dish. Also, we printed some simple structures at the beginning just to ensure that we were getting decent levels of cell viability as well as neuronal differentiation before getting too fancy with the actual structures themselves. So from that paper that I mentioned, one of the interesting things that we could do with the RX1, in case any of you have access to one, I think there is an RX1 at the NRC in Ottawa, and that group has been using it to print blood-brain barrier models, which is quite interesting. So what our group did was we actually used this, and this is one of the first times that bioprinting had been used to actually print hiPSC-derived neural aggregates. So you can see these aggregates, and then there are different ways of controlling them, so we could actually get them distributed in our tissue. So you could actually bioprint arrays of these neural aggregates, which is quite cool. And here's just some data showing that we maintain high levels of viability of our aggregates once they've been printed into our tissues, around 94% at day 10 and 65% at day 15. In both cases, we were just quantifying the fluorescence. And so I think we would also see a bit higher viability on day 15 if we had actually taken the cells out and done flow, as we've done this in later studies. Because the 2D nature of these photos isn't going to necessarily capture all of the effects in 3D as well. And we were able to culture these neural aggregates inside of our neural tissues for 41 days, which I think at the time was the longest anyone had actually been culturing any of these bioprinted neural tissues. And so that was quite exciting, and these tissues expressed mature neuronal markers, as well as astrocytes. So now that we had this good bioink formulation, next thing we were looking at, again, trying to cut down on the amount of labor students have to do and essentially mature our neural tissues in a quicker fashion, we began to investigate microspheres as bioink additives. And so, since I know a lot of you are CBS members, you're probably very familiar with the idea of microspheres in controlled drug delivery. Essentially, they're small spherical particles fabricated from biocompatible polymers. And you can encapsulate inside of them these small molecules and growth factors that I've talked about previously and use them to deliver different factors to promote the neuronal differentiation of iPSCs. And so our lab has done this for and characterized these microspheres for 3 different small molecules, including retinoic acid, glucosterone, and puremorphamine. And just to give you an idea of what the properties of these microspheres look like, so this is some of the photos and quantifying their microsphere diameter. This was really nice work from my PhD student who graduated last fall, Laura de la Vega. We made these microspheres relatively small, which makes it easy to incorporate into our bioink. And then we did some characterization of the release, and so they were consistently releasing the small molecule, in this case puremorphamine, over 48 days, which is a pretty nice extended time course and also corresponds to the time course for maturation of these iPSC-derived neural progenitors into neural tissues. And here's just a pretty image. So this was not bioprinted, but she actually incorporated these microspheres into these neural aggregates and then cultured them for extended time periods. And you can see that they really were effective. In this case, it was a combination of retinoic acid and puremorphamine releasing microspheres and making some nice and beautiful mature neural tissues. And so to sum up, a very nice paper, her puremorphamine releasing microspheres enhance hiPSC differentiation into chat-positive neurons, which is a motor neuron marker. She was trying to engineer spinal cords in this case. And this was in comparison to both positive, meaning soluble small molecules in the media, as well as negative controls where they did not receive treatment with these small molecules. And these engineered neural tissues that contain these drug-releasing microspheres actually did demonstrate electrical signaling after 64 days of culture. So Laura wanted to extend upon this work, and so we've been looking again at incorporating these microspheres into our bioinks. Another thing that we're really interested in is being able to place different types of microspheres where we desire inside of our bioprinted tissue, and here's just some proof of concept data. We were the second group to get the RX1, and so we wanted to show that it could do everything they claimed it was capable of. And if you just incorporate drug-releasing microspheres into your tissue, they look like what you see over here on your left. They look like little black dots. And so you can't really distinguish between 2 different types of drug-releasing microspheres. And so what we did was you can purchase these fluorescently labeled microspheres. And so we just used them to show we could co-localize the tissue, our microspheres to different portions of the tissue. So here's one fiber that contains the green microspheres, another with the blue microspheres, and then green again, with the idea that in the long term we would be patterning different types of drug-releasing microspheres in different regions of the tissue so we could print increasingly complex brain tissues. And again, here's just another slide showing, again, that we can layer. In this case, we're putting layers of microspheres on top of each other in a cross-hatching pattern. And so again, just showing we can put our microspheres where we desire them spatially within our tissue constructs. So this is some unpublished data from Laura's thesis. And one of the things that we've been interested in more recently is trying to show how consistent our constructs are, to try and show that it's a better technique than some of the other methods like organoid generation. And so here she was just measuring diameters and heights, and then the consistency of the weights. And she also found that between constructs using our bioink with no microspheres present and unloaded microspheres, you saw pretty similar weight distributions. And so bioprinting does seem to be producing relatively consistent constructs when we do this. And as I also mentioned, it's a relatively rapid process, assuming all your print heads and everything are working properly. Here's just some more showing these constructs. Again, she was printing these cylinders to resemble the spinal cord. We had two different types of drug-releasing microspheres here, pure morphamine and retinoic acid. So she was trying to replicate the effects she had seen in the aggregate tissues and our bioprinted tissues. And in this case, for the tissues, she added in both the red and green microspheres to show that they were co-localizing with the drug-releasing microspheres, and that we were getting even distribution of the microspheres throughout our tissues. The other thing shown here in C is she also stained this for DAPI. So this was neural progenitors that were essentially a single dissociated single cell suspension inside of our bioink. So we got really nice distribution across our tissues of the microspheres as well as the cells. And in this case, again, just as proof of concept, we were just using one concentration of each microsphere, and we wanted to actually get a uniform pattern. And then Laura cultured these bioprinted tissues, which consist of these hiPSC-derived neural progenitors with the drug-releasing microspheres for 35 days, and she actually did see some mature neural markers here. So beta-tubulin 3 is a neuronal marker, and so these are the different groups she had with no microspheres. This is a soluble control with pure morphine and retinoic acid in the media. The microsphere group that consisted of a mix of pure morphine and retinoic acid releasing microspheres, as well as an unloaded control. And so we actually saw neurons, HB9s associated with motor neurons, and so this was exciting. She was trying to make spinal cord tissue that contained motor neurons, as well as OLIG2, which is an oligodendrocyte marker. And then this is just what all the images look like merged together. And I don't show it here, but she also did some functional analysis of these tissues, and they did respond to stimulation with neurotransmitters by changing their action potential, which was quite exciting that we actually were producing tissues that could respond. So I think just to finish up some stuff, I'll talk a bit about our ongoing work. All the work I've talked to up to this point has been done using normal, healthy, human-induced pluripotent stem cell lines and neural progenitors derived from them. But where I think using hiPSCs has a lot of potential and a lot of power is using patient-derived iPSC lines. And so our collaborator at the University of British Columbia, Dr. Håkan Nygaard, he is an MD PhD and he runs their Alzheimer's clinic. And so he actually has his patients give blood and they're reprogrammed into these iPSC lines, which we can then characterize. And so we actually have several ongoing projects and grants where we are bioprinting neural tissues using his patient-derived iPSCs with the eventual goal of trying to test some off-label drugs. And so these are drugs which have been shown to be safe in humans, but they don't necessarily have a specific drug target. And so we actually have a few candidate drugs that we're really interested in trying to apply to our tissues and see if they can reverse some of the effects of Alzheimer's. So here's just some data from his lab where he has taken these blood draws from his patients and then reprogrammed the cells back into being iPSCs, which is what you see in A. And then in B, he's been able to differentiate these down into a cortical neural lineage. And so essentially, it's important to confirm that when you have some of these patient cell lines, you can actually get them to differentiate into the desired cell types you want. And we currently share two students. So my former honors student, Chris Lee, is currently at UBC, and he's active in the CBS student chapter there, working on this project and growing and culturing these Alzheimer's lines. And then in my lab, John Walter Schmolka has also done a lot of work, and I'm going to show some of his data here in a second. The other interesting thing that Dr. Nygaard's lab has done, in addition to showing that these iPSC lines make mature neurons, which is what you see in figure A, and astrocytes, they also become more Alzheimer over time. And so what that means is that these mutated cells show an increase in the expression of tau, which is what you see is characteristic of Alzheimer's disease. And then again, as these cell lines age, as the neurons get older in a dish, they also secrete more amyloid beta protein. And so the fact that you can track the changes in tau and amyloid beta is suggests that hopefully when we print our 3D tissues, they'll replicate these effects. And then if we can see that we can ameliorate some of these Alzheimer's symptoms using a drug, Dr. Nygaard, because he's a practicing doctor, could potentially prescribe these drugs back to his patients, which is very, very exciting, and also is very exciting in terms of personalized medicine applications. And so, as I mentioned, John in my lab, he has spent all fall culturing these diseased iPSC lines, which can often be difficult and finicky. And the last thing that he finished up before our lab was shutting down was getting some really nice images and collecting these bioprinted Alzheimer's tissues. So you can really start to see in them. I think you can really clearly see some of the projections that have been growing out. And so it really appears like we are making these three-dimensional tissues that could be disease models. Something interesting with Alzheimer's that both Hakon and other groups have shown is that you really need the different cell types present in brain tissue to fully replicate the phenotype. Just due to some of the cell therapy approaches, a lot of differentiation protocols result in just making a very pure population of either neurons or astrocytes. But there's a lot of evidence showing that the interplay between these cells is really determining some of these disease effects and the severity of these diseases. So future work, hopefully when things start to get back to normal and we can reopen again, we've been able to print these stem cell aggregates, and we have some interesting ideas going forward about how to use them as an aggregate array for screening drugs. Being able to precisely place multiple microspheres into different locations in our tissue is very exciting. And we hope to print more complex structures where we'd have different regions of microspheres and concentrations and combinations depending on the sort of tissue and the structures we wanted to make. The work bioprinting neural tissue models of disease is really exciting, and hopefully we will be getting some Parkinson's lines soon that we can characterize similar to our Alzheimer's line and start bioprinting those as well. And with Laura and Lila, who both have worked for me, we are co-founding a company called Axolotl Biosciences that will commercialize our novel NeuroBioInk. We already send it to a number of collaborators around the world, and there seems to be pretty significant demand. So hopefully we'll be able to start producing and shipping by the end of the year. And just to talk a little bit about why 3D bioprinted tissues for drug screening is beneficial, we can actually print a large number of tissues in a short amount of time that contain multiple cell types. I didn't show it today, but we have some very exciting work with our 3D cancer models where we show that you can observe effects for different drugs in our 3D models that you don't see in 2D culture, which suggests they're much more relevant as a tool for drug screening compared to using just standard 2D cultures. We've got some really nice data showing that we can recapitulate the functionality of our neural tissues in vivo. And I think one of the most exciting things is the potential for personalized medicine, both for Alzheimer's, Parkinson's, as well as potentially we would like to get some patient glioblastoma samples in that we could also start bioprinting into our tissue models as well. And one last thing that I've been thinking about lately, and probably other people have with the COVID-19 virus, there's evidence of neurological effects caused by COVID-19, including it's very commonly reported that you lose smell. There's also headaches, nausea, vomiting, and seizures. A high percentage of even young people, I think, who've suffered from this end up having strokes. There's also a higher incidence of epilepsy. And so there's evidence, a study out of China showed that the virus does infect neural tissue, and there's some interesting hypotheses out there that potentially this virus is being transmitted in the brain via synapses. And so I think there's a lot of interesting biology there. They did, I think GlaxoSmithKline just released a preprint where they've shown, they've published how to make the pseudotype virus, which can infect but is not replicate. So we're hoping when things get a bit back to normal, that it would be really interesting to study how the virus does affect neural tissues, figuring out where the ACE2 receptor is, what cell types it is expressed by, the levels of expression, and what actually gets infected. And so I'm happy to talk a bit about that as well. And then just two last things for people who might be bored or are looking for places to submit their work. I'm on the editorial board of Cells, Tissues, Organs, and they are currently accepting submissions for their Young Innovator issue now. And it's also a good place if you want to send any of your review articles. I'm also an editor at STEM Journal, which again, another open access journal. And just again, if you are writing your reviews or looking for places to submit your research, it's another good resource. It's open access, and so it's been a good resource. I published in their equivalent when it was called STEM Book back when I finished my PhD in 2008, and then I updated it with my PhD supervisor, Shelley Sakayama Elbert, last year, which was one of their first published papers, was a reprinting an updated version of our book chapter on how to combine stem cells and biomaterial scaffolds for constructing tissues and cell delivery. And so it's a good resource, and also if you're just interested in reading what's out there, it's great. So with that, couldn't do any of this without my lab or funding agencies. We're very excited to be funded by INSERC, the Michael Smith Foundation, the Pacific Parkinson's Research Institute, Canada Research Chairs, Alzheimer's Society. We have exchange grants with Brazil and Texas, even though that probably won't be happening for a while in my tax. And then Laura De La Vega, Ruchi Sharma, Megan Robinson, Chris Lee, Kwong, Nadia, John, Kieran, Callie, Josh, Alana, and Dimitri. I'll take questions from Hallie.
Perfect. Thank you very much, Dr. Willerth, for a very interesting and informative presentation. For any of you who've joined late, you can send your questions to me by a private chat, and I will ask Dr. Willerth your questions. For a first question, we have a question from Irene Guzman Soto from she's a master's student from the OHRI, the University of Ottawa Heart Institute, and she is asking, how is the fact that there are morphological changes in neurons after stimulation addressed in 3D bioprinting, if there's also a required distance between them for a successful synaptic transmission?
That's a good question. So we've been mainly printing neural progenitors, and we have played a bunch with cell densities, and I didn't show it today, but my students have done work optimizing the density, because that's a very important factor when you're bioprinting, is the number of cells, and you have to balance it depending on your bioink and things like that. And then just in terms of getting the proper distancing, I think it's, a matter of, with the neuroprogenitors, it's a bit different, because then they sort of mature and differentiate. And I find that depending on your cellular populations, you'll sort of get a balancing out effect, or if you have a certain number of neurons, then some of your stem cells will then become the support cells, sort of maintaining the ratio they need. We have bioprinted some motor neurons, which did retain their functionality from BrainXL, which we thawed them, and then I think a day after they were still showing synaptic function. So yeah.
Perfect. Thank you. The next question is from our director, actually, Dr. Emilio Alarcon. He is asking, one of the limitations in 3D printing is the numerical methods that are used for replicating the 3D structure of the target tissues and organs. How do you see this area of research evolving in the field of 3D bioprinting of neural tissues?
Oh man, I keep asking all of our collaborators, it's getting sunny here. I keep asking for good software for predicting if a structure will print. It's such an interdisciplinary field, and then making a material printable is still sort of trial and error, and even making structures for our cancer work, where we're doing co-culture, where you have two very, we do a lot of preliminary testing, usually with food coloring, in order to ensure that we can actually print the structure that we want to make. And so that's one of the areas I really think is important, is being able to consistently predict, based on a CAD file, will the structure print and will it maintain its shape? Because I know that's something that my students have worked a lot on, and we haven't gotten better than essentially trial and error. But I also tell Aspect that I'm like a computer scientist.
Perfect. The next question is from Dr. Mo Al-Khalaf. He's a postdoc at the University of Ottawa Heart Institute. His question is, when it comes time to drug delivery, is there any thought regarding bioprinting the drug body pathway that it goes through, an oral drug would go through the digestive tract, liver, bloodstream before reaching the heart, for example. Any thoughts on about expanding the type of cells, tissue, bioprinted to create a more lifelike trial?
Yeah, so I had mentioned Pirellis earlier, and we had gotten some of their vasculature structures. So I think how they normally work is they have, it's like a 3D printed sort of plastic structure that makes vasculature. And so my co-op students, we actually 3D printed our tissues onto these vasculature baskets, because ideally that's what you would eventually want. There are some really cool, I think it's Emulate, who does microfluidic stuff, where they actually have a really cool blood-brain barrier chip that really does a good job of modeling both the blood compartments and the brain compartments. Some of the difficulties you run into when you're trying to do these co-culture models is finding a cell culture media that keeps both tissues happy too. But yes, we do think about that.
Okay. The next question is from Marcelo Munoz from the University of Ottawa Heart Institute as well. He's asking, what is the best resolution you can get with the best 3D bioprinters on the market now?
Oh man, let me check what ours was. We were, let's see, I'm trying to think. It also really depends on the bioink too, because I think for some of the things like alginate, you get a more reproducible structure, but it doesn't support cell growth as well. And so you're sort of trading off your accuracy for cell function. Okay, Laura's applied science paper. And it also depends a bit on how you bioprint and the degree of infill and so forth. So let me see. Going to quickly, I was like, I know I made and for graduate students calculate all of the sizes of our constructs. So let's see, yeah. So we were, our fibers tend to be 176 microns plus or minus 10 microns. So pretty decently accurate considering we're working with fibrin, which is much more sticky.
Perfect. A next question from Amanda Juraski from the University of Sao Paulo in Brazil. She says, great presentation, Dr. Willerth. My question is regarding bioprinted scaffolds for spinal cord, which technique stands out? And she's a PhD student at USP.
Nice. Hey, hello, Brazil. Obviously I'm partial to my work, but Michael McAlpine's group had some really cool patterning. I think it was an advanced functional materials paper in 2019, where they did some really nice structured, where they were printing tracks of neurons, then tracks of oligos, all in the same construct. And so I thought that was really impressive. They were getting both the co-culture and it was a very detailed spinal cord-like structure. So that was one of the papers that I was really impressed with. And his group has been doing a lot of really interesting work, not just on the tissue scaffolds, bioprinting tissues, but also they've done a lot of actually 3D direct printing of electronics for neural prostheses that where you can 3D print electronics onto skin.
Very cool. So I have a question as well. I'm Hallie. I am doing my master's at the University of Ottawa, also working a little bit with neural cultures. So my question is, you showed a lot of fluorescent images. Have you tried to deposit your bioprinted tissues on microelectrode array substrates to look at electrophysiological signature and data from the neurons?
Yeah, so we got with the non-bioprinted tissues for Laura's paper, one of the reviewers asked, and we did get to borrow one of the Iaxion Maestro arrays, and our collaborator has one as well. And so we didn't print tissues there. We actually just cultured them on the arrays to measure the signaling, and it was great and wonderful. Then we had to send the instrument back. We do use flipper dye a lot, which people have been using to measure, and it doesn't give you the individual cell levels, but it does measure the overall responses of the tissues. So that at least gives you some idea that the tissues do respond to neurotransmitters. I know our collaborator does have one of the microelectrode arrays, and so we've just been sort of talking about which way do we want to, how do we want to get our tissues from Victoria to Vancouver and vice versa. And also, Dr. Nygaard recently got one of the bioassembly bots, which is very, very cool. That has the like 6-axis bioprinter. I know my lab went over and got trained on it, and they were all very jealous. And also, apparently, it changes your cell culture media for you, which things I can't compete with.
Perfect. We have another question that just came in. It's a PhD student at a technical university in Turkey. Ijiken Sumbeli, I apologize if I didn't pronounce your name right. He says, or she says, thank you for your presentation, Dr. Willerth. I'm a PhD student in Turkey, studied biofabrication during my master's here. I looked for neural tissue biofabrication programs and couldn't find much. People tend to work with other types of tissues in biofabrication. Why do you think that this happens?
Neural cells are really finicky. I think before Michael McAlpine and Laura's paper came out in 2019, the best cell viability you're getting if you bioprinted any stem cell derived neural cells was 40 to 50%. And the other thing is it's just sort of the biology is pretty complex, and then a lot of the stuff with the bioprinting is like they would print cells and then, you know, characterize viability like 3 days later. And they're like, yes, we made a neural tissue, which is not really going to tell you much about function. You can see that, I think you see that in bioprinting. Also microfluidics, where you have some things like neural cells, which are very hard to culture. And so people just don't either want to do it or spend the time. I think you see it also with cardiac too, because it takes a lot to get your cardiac cells to mature and stay mature. And just the way I think a lot of bioprinting has evolved is it has come from more of a mechanical, electrical background. And so there's been less of an emphasis on the materials, which is one of the reasons my group is very interdisciplinary, is because it's important to have some of these more sophisticated materials if you're going to want to print complex tissues. And I do hear this from especially the cell biologists who are like, I hear how bioprinting is really great, and then they go to print their stem cells and half their cells they've just spent 60 days culturing die. And so they get very frustrated with the technique.
Mm-hmm. For a final question here from our director, Dr. Emilio Alarcon, he says, what do you think of the use of bioreactors in neural tissue?
Oh, I think there's actually some really cool progress being made. It's weird our lab hasn't published on organoids, even though we've made them. One of my colleagues who's at Vanderbilt, Ethan Lippman, his group has made a really cool, as I mentioned with organoids, reproducibility has been a big issue. And so they made, I think, an open source bioreactor scheme in order to essentially make little individual chambers to make it more consistent when you're making your organoids. So there's a lot of really cool work out there, especially in terms of the organoid stuff, where bioreactors play a very crucial role. And I think definitely, I think if we go towards making more developed cell models, like especially if you want to have like a vasculature, that's going to be something you'll really have to consider. Even if it's not a bioreactor, is some way of flowing your blood through your tissue.
Mm-hmm. Perfect. Well, I think we'll have to wrap up here. Thank you very much, Dr. Willerth, for joining us today and for an amazing talk.
Yeah. Well, thanks everybody, and have a good week.
Yes, this presentation has been recorded and will be available on the BEaTS Research Radio website if you'd like to take another look at it. And make sure to join us next week for a presentation by Dr. Ali Kerem Hosseini, who will be speaking to us about engineering and precision medicine. So thank you to all of our listeners, and again, Dr. Willerth, for joining us, and we'll see you all next week.